Hello, welcome to Biology, the scientific study of life. Regulation of kidney function. One of the most important aspects of the mammalian kidney is its ability to adjust both the volume and osmolarity of urine, depending on the animal's water and salt balance and the rate of urea production. In situations of high salt intake and low water availability, a mammal can excrete urea and salt with minimal water loss in small volumes of hyperosmotic urine. But if salt is scarce and fluid intake is high, the kidney can get rid of the excess water with little salt loss by producing large volumes of hyperosmotic urine as dilute as 70 mosm each liter compared to about 300 mosm each liter for human blood. This versatility in osmoregulatory function is managed with a combination of nervous and hormonal controls. One hormone that is important in regulating water balance is antidiuretic hormone, short-term ADH. ADH is produced in the hypothalamus of the brain and is stored in and released from the posterior pituitary gland, which is positioned just below the hypothalamus. Osmoreceptor cells in the hypothalamus monitor the osmolarity of blood when it rises above a set point of 300 mosm each liter, perhaps due to water loss from sweating or to ingest of salty food. More ADH is released into the bloodstream and reaches the kidney. The main targets of ADH and the distal tubules and collecting ducts of the kidney, where the hormone increases the permeability of the epithelium to water. This amplifies water resorption, which reduces urine volume and helps prevent further increase of blood osmolarity above the set point. By negative feedback, the subsiding osmolarity of the blood reduces the activity of osmoreceptor cells in the hypothalamus, and less ADH is then secreted by only the gain of additional water on food and drink can bring osmolarity all the way back down to 300 mosm each liter. Conversely, if a large intake of water has reduced the blood osmolarity below the set point, very little ADH is released. This decreases the permeability of the distal tubules and collecting ducts, so water resorption is reduced, resulting in increased discharge of dilute urine. Increased urination is called diuresis, and it's because ADH opposes this state that is called antidiuretic hormone. Alcohol can disrupt water balance by inhibiting the release of ADH, causing excessive urinary water loss and dehydration, which may cause some of the symptoms of hangover. Normal blood osmolarity, ADH release, and water resorption in the kidney are all linked in a feedback loop. This or that contributes to homeostasis. A second regulatory mechanism involves in a specialized tissue called the juxtaglomerular apparatus, JGA, located near the afferent arteriole that supplies blood to the glomerulus. When blood pressure or blood volume in the afferent arteriole drops, for instance, as a result of reduced salt intake or loss of blood, the enzyme renin initiates chemical reactions that convert a plasma protein called angiotensinogen to a peptide called angiotensin second. Functioning as a hormone, angiotensin second raises blood pressure by constricting arterioles, decreasing blood flow to many capillaries, including those of the kidney. Angiotensin second also stimulates the proximal tubules of the nephrons to reabsorb more NaCl and water. This reduces the amount of salt and water excreted in the urine and consequently raises blood volume and pressure. Another effect of angiotensin second is stimulation of the adrenal glands to release a hormone called aldosterone. nephron, distal tubules, making them reabsorb more sodium in A positive and water and increasing blood volume and pressure. In summary, the renin-angiotensin-aldosterone system RAAS is part of a complex feedback circuit that functions in homeostasis. A drop in blood pressure and blood volume triggers renin release from the GJA, or in turn the rise in blood pressure and volume resulting from the various actions of angiotensin II and aldosterone reduce the release of renin. The functions of ADH and RAAS may seem to be redundant, but this is not the case. Both increase water absorption, but they counter different osmoregulatory problems. The release of ADH is a response to an increase in the osmolarity of the blood. As when the body is dehydrated from excessive water loss or indicates intake of water. However, a situation that causes an excessive loss of both salt and body fluids, an injury, for example, or severe diarrhea, will reduce blood volume without increasing osmolarity. This will not induce a change in ADH release, but the RAAS will respond to the fall in blood volume and pressure by increasing water and Na positive reabsorption. ADH and the RAAS are partners in homeostasis. ADH alone would lower blood Na positive concentration by stimulating water reabsorption in the kidney, but the RAAS helps maintain balance by stimulating Na positive reabsorption. Still another hormone, a peptide called ethereal natriuretic factor ANF opposes the RAAS. The walls of the atria of the heart's release ANF in response to an increase in blood volume and pressure. ANF inhibits the release of renin from the GJA, inhibits NaCl reabsorption by the collecting ducts, and reduces aldosterone release from the adrenal glands. These actions lower blood volume and pressure. Thus, ADH and RAAS and ANF provide an elaborate system of checks and balances that regulate the kidney's ability to control the osmolarity, salt concentrations, volumes, and pressure of blood. The precise regulatory role of ANAF is an area of active research. The flexibility of the mammalian kidney enables to adjust rapidly to contrasting osmoregulatory and excretory problems. The South America Vamba bat illustrates this versatility. Bats of this species feed on the blood of large birds and mammals. The bats use their sharp teeth to make a small incision in the victim's skin and then lap up blood from the wound. Anticoagulants in the bat's saliva prevent the blood from clotting, but the prey animal is usually not seriously harmed because Vamba bats often search for many hours and fly for long distances to locate a suitable victim. They benefit from consuming as much blood as possible when they do find prey, so much that after feeding, a bat could be too heavy to fly. However, the bat's kidney offload much of the water absorbed from a blood meal by excreting large volumes of dilute urine as it feeds up to 24% of body mass per hour. Having lost enough weight to take off, the bat can fly back to its roost in a cave or hollow tree where it spends the day. In the roost, the bat faces a very different regulatory problem. Its food is mostly protein, which generates large quantities of urea, but roosting bats don't have access to drinking water to dilate it. Instead, their kidneys shift to producing small quantities of highly concentrated urine, up to 4,000 600 mosm each liter. An adjustment that disposes of the urea load while conserving as much water as possible. The Vamba bat's ability to alternate rapidly between producing large amounts of dilute urine and small amounts of very hyperosmotic urine is an essential part of its adaptations to an unusual food source. These different or diverse adaptations of the vertebrate kidneys have evolved in different environments. Notice that vertebrate animals occupy habitats ranging from rainforests to deserts and some of the saltest bodies of water to the dilute waters of high mountain lakes. Variations in nephron structure and function equip the kidneys of different vertebrates for osmoregulation in their various habitats. The adaptations of the vertebrate kidney are most clearly revealed by comparing species and inhabit a wide range of environments, or by comparing the responses of different vertebrate groups to similar environmental conditions. Okay, thanks for listening.